Welcome back to Bio 111. This is chapter 15. We're going to be looking at genes and proteins. All right, I want you to recall from earlier chapters when we talked about proteins, that proteins are made up of amino acids. Right? In the world of proteins, if a protein is a word or a sentence, the amino acids are the letters. And by making different combinations of amino acids, we get proteins with different properties, right? If you see these blue boxes that are surrounding some parts of these amino acids, that is, those are the parts of these amino acids that are the R groups that are varying the properties for these molecules. And that is what gives the versatility of these proteins. It allows them to do so many different things. All right, I've mentioned this a few times, the central dogma. We talked about it last chapter. DNA, that's where all the data in the cell is stored in life. It's all in DNA. Now, the information from DNA flows from DNA to RNA, specifically to mRNA, messenger RNA, and then into proteins. So DNA is first transcribed into mRNA because we're not changing the language, right? We're going from DNA to RNA. We're transcribing it. And then from mRNA, we translate into proteins because we're going from a nucleic acid to a protein. So it's now a new language, so we call it translation. All right. Instructions on DNA are transcribed onto the messenger RNA. Ribosomes are protein and RNA made machines that read the genetic information stored on the mRNA, and they translate it into proteins by attaching amino acids together based off of the sequence in the mRNA. Now, if we go back and go to the previous slide, this bottom picture, we see that each of these mRNAs, these little sticks, EPA, these are mRNAs, sorry, these are tRNAs, they read the DNA in 3 base pair combinations. We call those codons. All right? So it takes 3 base pairs, AUG in this case, or UUU, CGA. Notice there's no T's in here because we're looking at RNA. In RNA, we replace the thymine with uracil. These codons match an amino acid. Now, the way this works, there are 64 possible codons that code for a total of 20 amino acids, and 3 stop codons that tell this machinery when to stop adding to the protein, the polypeptide chain. All right. Now, this is sort of the cheat sheet for determining what amino acid you're going to get given the sequence of DNA, the 3 base pair sequence of DNA we're calling a codon, right? So all you have to do is look up what 3 letters you have, what 3 base pairs you have, and you can match it to the amino acid. All right. So we'll get to a few things here about mutations. We talked about mutations in the previous chapter, but what does a mutation really do? Why is a mutation, why does a change in genetic code, why does it really cause any sort of issues with a phenotype or with physiologically with the living organism. And what it comes down to is it changes the way that DNA is transcribed and then later on translated into a protein. All right, so a frame shift mutation, that's the deletion of two nucleotides that shifts the reading frame of an mRNA and changes the entire protein message, creating a non-functional protein or terminating protein synthesis altogether. Now, we're going to go back to 2 slides here. A reading frame, when we talk about a reading frame, it refers to which nucleotide starts the first codon. For each segment of DNA, there are 6 possible reading frames. All right? So, what do we mean when we say a reading frame? Well, if you recall, we are reading this, or the ribosomes are reading these sections of RNA, of mRNA, 3 base pairs at a time, these codons. Now, if we go and look at this, let's say that we've got a sequence and it's UUUC, right? So this top left corner doesn't change anything. But let's say that it's UUUA. We lose one of the U's, and suddenly, instead of being the PHE you see right there, which is a particular amino acid, we're now LEU, right? We shift the reading frame forward, causing a different protein to be made. And likely the protein will not work, all right? All right, so that's it for section 1. Come back in for section 2 in the next video.